Hello everyone, and welcome again to A Pint of Astrobiology, the show where I discuss the latest conundrums in the field of astrobiology over a pint of beer. My name is Ben Pierce, I am an astrobiologist, an amateur brewer of beer, and your humble host. And today I'm drinking a Belgian Saison that I homebrewed. It's typically a highly carbonated style brewed in Belgium that has generally fruity and spicy notes. This one's just peachy. It's only peachy. That's the note I get. In today's episode, we're going to be discussing detecting life on the closest potentially habitable body in our solar system, and my favorite planet, Mars. And also, today we have a special guest on my show, so be sure to stay tuned for that. Mars has an average surface temperature of minus 63 degrees Celsius or minus 81 degrees Fahrenheit, which is similar to the average temperatures in Antarctica. But, like in Antarctica, Mars has hot spots where you can find liquid water. The most notable of these are the subsurface lakes that were recently detected in the South Pole region of Mars, detected by the European Space Agency's Mars Express orbiter by radar measurements. Now, the jury's still out as to whether these are true lakes or just maybe pockets of liquid water in saline ice, but still liquid water is present, therefore we can fathom the presence of life in these regions. But how do we think, or why do we think, that life can survive deep below Mars' surface in regions such as this? Well, take Lake Untersee, for example, a seasonally ice-covered freshwater lake in East Antarctica. Now, at the bottom of Lake Untersee are these large photosynthetic microbial mats up to half a meter high. Microbial mats are these multi-layered structures of microorganisms, which you can see in better detail here. These are analogous to some of the first life forms on our planet, which left their mark in the fossil record dating back to 3.7 billion years ago. And you can see those structures here. But how do these microorganisms survive in such nutrient-poor, low light, and very cold conditions? Well, they've evolved to become scavengers and recyclers. Reduce, reuse, recycle. They get whatever nutrients they can from the water column below and from the sediment above, and they make do. And they also get about 0.1% light transmit through the ice and the water above to the bottom of the lake where they survive. But that isn't even that crazy, because there are organisms that live 2.5 kilometers underneath Earth's surface where there is absolutely no light. So how do those organisms survive? So to talk about this, I'd like to introduce my colleague and friend, Sean Ford, who has found microorganisms living 2.5 kilometers underneath Earth's surface. Take it away, Sean.
That's right, Ben. Me and my colleagues have found microorganisms living deep within the surface of the Earth. These organisms are extremophiles, meaning that they live in conditions that to us seem really, really harsh. These microbes like super salty water. I'm talking 30 times saltier than the ocean. They eat things like iron and sulfate, and some even live off of only gases like carbon dioxide and hydrogen. And all they need for life are those two things and the water that is trapped within the crust of the Earth.
Thanks, Sean. Cheers. So, how are we to detect life on Mars if it is potentially kilometers underneath Mars's surface? Well, that's the challenge. One simple way would be to detect the biosignature gases emitted by these microbes. Remember back to episode 1 of this series, and scientists have done just that. The Curiosity rover has detected 21 parts per billion of methane near the surface of Mars, which is only 100 times less than the methane in Earth's atmosphere from our methanogenic microorganisms. So that's pretty exciting. But we can't say for certain whether this methane is actually from methanogenic Martian microbes or if it's simply from water rock interactions. We need a more compelling result. Probably the most promising way to detect past life on Mars is to have a sample return mission. The Perseverance rover, which is on Mars right now, is going to collect samples from regions of interest for later pickup and delivery back to Earth. So perhaps we'll be able to see some visual evidence in the samples of past life. Things such as the fossils left behind from ancient cells, as you can see here, or maybe the layered structures left over by ancient microbial Martian mats, as you can see here from analogs from what we see on Earth. And if there's no visual evidence, maybe scientists will be able to detect biomolecules that have one of the characteristic signatures that tells us that those biomolecules were produced by life and not some sort of non-life. Things such as being low in carbon-13, the isotope, or having all one-handedness, either left or right, for the molecules that have handedness, such as amino acids and sugars. And you need all one-handedness for life because these things need to chain together to make things like proteins. And you can't have mixing hands all about. However, the plan for a Mars sample return mission is still in early development, and with the way the space race is going, human astronauts may arrive at Mars and collect their own samples before any robotic mission can come collect those cached samples from Perseverance. If we're going to find life on Mars, we're likely going to have to dig into the subsurface where liquid water is. But the real question, the ethical question is, should we do it, knowing that we could be potentially destroying the habitats of alien life forms? I'm going to leave this as a cliffhanger for now. And in my next video, we'll dive into this highly controversial topic known as planetary protection. Oh, I'm slurring my words now. That's all for now. I almost got the last sip. One sec. Cheers.